Okay, so last one for plasma membranes. We've got a little bit more to cover as far as active transport is concerned. So let me get over to that screen. So now we're going to talk about bulk transport. And so what this is telling us, sometimes we need to carry a lot of stuff, or we need to carry one really big thing, and that doesn't fit through our carrier proteins or our transport proteins or our channel proteins or what have you. So we need to have a different mechanism for carrying a high quantity or one large thing or a few large things. Okay, so what you want to know is what is endocytosis and how like it comes in its all these different flavors. And then exocytosis, which is the opposite of endocytosis. So, okay. So to your root word dictionary, you need to add endo, which means inside. Exo means outside. Cyto means cell. Phago means to eat. And pino means to drink. And if you can understand, get these, then you can start to just kind of logic out and understand what is all these other crazy, scary, intimidating words mean if we know the root words. Okay, so endocytosis is just a mechanism for moving large molecules, parts of cells, maybe even a whole entire cell into another cell. So there's lots of different varieties of this, and we will talk about each of those. But basically all endocytosis is what we have is this little pocket. This plasma membrane starts to pinch together a little bit and starts to form a little pocket. And then whatever it is that the cell is trying to move migrates into that pocket, and eventually the plasma membrane will pinch off. And now we have this little vesicle inside of the cell that contains whatever that thing is that the cell wants to move. So basically, these are all the things that we're going to talk about are variations on this process. Okay, so the three flavors of endocytosis are phagocytosis, pinocytosis, and receptor-mediated endocytosis. So let's talk about each of these. Phagocytosis means cell eating. This is where the cell takes in large particles and can be even other cells. In fact, that's what some of our white blood cells, specific types of them, do is create these little pockets here, and they collect the microorganism that wants to, the virus or the bacteria or what have you, that wants to cause some problems in our with our bodies, and they say and then they whatever the particle is trapped inside of the cell membrane, and then this vacuole will go and move to an organelle called a lysosome. And the job of the lysosome, and if you remember hydrolysis, you might remember that lys means to break. So this is the body that breaks stuff. This is the barbarian of the cell, and just like, I'm going to smash you with a hammer. Okay, so the vacuole merges with an organelle called the lysosome. The lysosome will break whatever that particle is. Okay, and then pinocytosis is very similar, but this means cell drinking, because the guy that discovered it thought that cells were just using this to bring in a lot of water in one go. And it does bring in water molecules this way, but it can also bring in other stuff. And so in this diagram, I don't know what this is supposed to indicate, maybe a sugar. But the differences between pinocytosis and phagocytosis is that the pinnos, the vesicles or the vacuoles are tend to be much smaller in diameter, and they don't necessarily merge with a lysosome like they do in phagocytosis. And sometimes even the cells will like grab some stuff, do this pinocytosis, and then take it all the way through the cell and then like loop it out on the other side. And then when that is happening, we call it transcytosis, which just means we're taking it, we're transporting it through the cell, and then we're giving it, putting it out into the extracellular matrix on the other side of the cell. Okay. And then this receptor-mediated endocytosis is pretty much the same thing. The difference is that we have either some glycoproteins or some glycolipids that have an affinity for a certain substance. And so that certain substance will come and dock onto those receptors, and then we still have this process of it getting pinched off and then put wherever it needs to go, whether that's to the lysosome or out the other side of the cell. What is tricky is that there are some viruses and bacteria that can use this mechanism. I am trying to say two words at once. Mechanisms or methods. So viruses and pathogens might come and enter the cell using this mechanism. So they might have adapted similarities between the substance that the cells want, and then they coat their body, their cells in that, and then our cells bring those in to our cells thinking that we're just trying to translocate something, and then we're like, ah, crap, now we're sick. Okay, so that's receptor-mediated endocytosis. Big difference is that we have receptors. And then exocytosis is the last form of active transport that we're going to talk about. And then this is just basically taking out the trash. This is getting rid of stuff that we don't want. So maybe after the lysosome lysed whatever they needed to lyse, now we got to like take all that stuff out and get rid of it. So what happens is that the waste products will become entrapped with a plasma membrane. That vesicle will move toward the exterior of the cell. This plasma membrane will fuse with our cell's plasma membrane, and then dump the trash out into the extracellular fluid. Okay, and then I've put this in the PowerPoint for you guys as just an ease for you. So this is taken from your textbook. This is just to help you remember the different ways that stuff is transported in cells, whether it's active and passive, and what's moving, and how stuff is going on. So use this as a nice little summary study guide. Okay, so that is everything that I have for you as far as plasma membranes go, and cell transport. We will be doing a lab on Thursday, so make sure that you come to class on time and are ready to do lab stuff for about 2.5 hours. Okay, I'll see you guys on Thursday.